Yeah. So I request Professor Rajesh Sundaresan to please chair the session. Thank you.
Yeah. Hello, everyone. Welcome back. I would like to welcome Andrew back to the stage once again, this time to answer questions that you have posed. We'll follow the same procedure where we'll go through the questions that were asked, that were posted, for which Andrew may have some possible answers and thoughts. Those who have questions, please post them in the Q&A instead of in the chat, in the Q&A, and we will keep track of that. So we'll begin with a question that has been asked for lecture 13. In molecular MIMO containing 2 receivers, suppose we are transmitting n molecules. We assume that the rate of TX RX1 channel is fixed, say R1. Basically, by fixed rate, we mean that we are first ensuring communication between the TX RX1 channel and analyzing the TX RX2 channel. Then is it proper to assume that the leftover molecules, say K out of n molecules, that arrive in the TX RX2 channel is dependent on the TX RX1 channel?
Okay, so this question is related to a question that we had last time about the independence of arrivals at receivers. And I have to say that after thinking about it and discussing with one of the students yesterday, I realized that what I said yesterday was incorrect. So in fact, there is dependence. Even if the Brownian motions are independent, there is dependence between the receivers. So it is correct, as in this question, to say. In fact, let me share the question so that everyone can see it. It is correct, as in this question, to say that there is dependence between the receivers. And in order to think about why there is dependence, imagine the following scenario. So suppose there are exactly 2 molecules, at least 2 molecules, and there are 2 receivers. So if I observe both molecules at one receiver, RX1, then I know for certain that there will be 0 molecules observed at RX2. But in general, there are not 0 molecules observed at RX2 as a marginal distribution. Therefore, there must be dependence. It can't possibly be independent. So there is definitely a dependence between the receivers, even if Brownian motion.
Okay, we'll go to the next question. Can the mutual dependency of absorbing receivers be effectively utilized to reduce ISI? For example, in a point-to-point link with an absorbing receiver, can the proper placement of another absorbing receiver reduce ISI?
I think this is a very interesting question. I had actually never thought about this before, and when so I received these questions about 2 hours ago. I was really struck by how interesting this question is, because I don't think anyone has actually looked at this, whether it's possible to design the environment and the locations of the receivers geometrically to ensure that molecules are absorbed so that ISI is reduced. I suspect that it is possible. I'm not aware of anyone who has ever looked at this. I think it's an intriguing idea, and whoever asked this question, I would encourage you to follow up on it. I think it's a potentially promising line of inquiry.
Thank you. The next question is, don't we need a central node in molecular MIMO to combine the data received by each of the receivers and do further processing?
Yes. So in any MIMO system, you require a central node to complete the processing and the data fusion. So that's certainly true in MIMO. If I didn't say that in the lecture, I certainly meant to. But all of the receive, I mean, they're not antennas, but let's call them antennas for argument's sake. All of the antennas are connected to a common data sink. That data sink does collective processing. So the answer is yes.
Thank you. Next question. As the noise term is date dependent, Can a sparse coding technique significantly reduce ISI? I think it's probably rate dependent.
Yeah, I think they mean data dependent. I
That's-
think
data dependent, yeah.
Data dependent.
Yeah, I think that's right. By sparse coding technique, I assume you mean something like pulse position modulation, where there is there's a lot of time between pulses that are transmitted. I think that's probably the right idea to reduce ISI. That said, it's known that a pulse position modulation technique like that reduces the data rate quite a bit. Having said that, ISI is a severe problem, as we've seen throughout these lectures. So I think this is something that you might try in order to reduce the ISI.
Thanks. We'll now move to questions in lecture 14. How does the binding of a ligand to the receptor trigger secondary reaction in the receiver? What are the results of these reactions?
Okay, so this is a really nice question. You may recall, and I'm actually, now that I think about it, I'm not sure that I made this link explicit in those lectures, but you remember an earlier lecture when we first started talking about the Michaelis-Menten kinetics. You will recall that we talked about a substrate binding to an enzyme being processed, and then the process substrate being released. So this is the process by which this is how I think about the secondary reactions occurring. So what happens in signal transduction is that a ligand binds to a receptor, and then the receptor undergoes a conformal change. We interpret this as a chemical reaction, and we describe it using the master equation, as we talked about in the lectures. What happens then is, as a result of that conformal change, further chemical reactions occur inside the cell. So the signal is transduced across the cell membrane, and this is why we call it signal transduction. Within the cell, different chemical messengers called second messengers are produced, and it's those second messengers that actually produce the required result inside the cell. So the mechanism by which signal transduction produces its results was noticed about 50 years ago, or more than that, probably 60 years ago, in the 60s. And what it was noticed was that the messengers had messenger chemicals like hormones throughout the body have diverse impacts on all kinds of different populations of cells. And the question was, how is this possible? How could it be that one messenger could result in all kinds of different effects on all kinds of different cells? And the answer was because each type of cell had its own receptor and was producing different second messengers inside the cell. So this observation actually led to, I think, more than one Nobel Prize in medicine. But the secondary reactions occur in much the same way we talked about in the Michaelis-Menten reactions. So substrate attaches to enzyme. The enzyme processes the substrate into a second product. That's basically the idea.
Okay, so we have the next question. What is the advantage of discretizing the master equation?
Okay, good question. So on the one hand, I would say it's a matter of preference. If you are more comfortable dealing with continuous time, then you can leave the master equation in continuous time. If, like me, you think your mind works better in discrete time, then discretizing the master equation helps you visualize the system a little better. It does go a little further than that, because discretizing the master equation transforms the system into a discrete time Markov chain, and we have different results. So I'm not going to say more results, but we certainly have different results for how discrete time Markov chains work, and we can use those results to develop closed form results about the progress of the chemical reaction. One example is, I'm not sure if we directly talked about it in this series of lectures, but one example is we know a lot about how discrete time Markov channels work. So there's a ton of work in information theory about how discrete time Markov channels function, what their information theoretic capacity is, what their capacity achieving distributions are, and in some of my work on signal transduction, we've actually used those results to establish the information theoretic capacity of a very simple signal transduction system. So the advantage would be in that sense to give yourself access to results, useful results and useful tools having to do with discrete time Markov chains.
Okay. We'll move to the next question, lecture 15 now. As ISI is a major issue in MC, Markov channels, can we model it as an information channel with having memory?
Definitely. So we've established throughout these lectures that, for one thing, there is ISI. ISI is a problem. How the memory works is, it depends on exactly how the channel is modeled. So we've seen, on the one hand, for non-absorbing receivers, we've seen a Markov, excuse me, not a Markov, a linear system model, a concentration model, in which we've derived the form of the impulse response. So given the impulse response, we can use techniques from conventional communication to filter the impulse response and potentially eliminate the ISI or deal with it in other appropriate ways. So that would be an example, conventional example of molecular communication as an information channel with memory. However, in other circumstances, the memory structure is more complicated. So we talked also about this problem of information molecules arriving in not the same order they were transmitted. So that's also a kind of memory, but it's one that is not well modeled by conventional techniques for dealing with ISI. And in fact, it's a very difficult issue because as we've talked about over the previous Q&A sessions, what you have to do is, or it seems like the most simple solution would be to sum over all the possible permutations of the arrivals. And that's computationally intractable. So yes, it is definitely an information channel with memory. Depending on how it's modeled, it can be handled either with conventional techniques or potentially new techniques, which are currently unknown and an open problem, would have to be derived.
Thank you. Next question is about definition of channel capacity itself. In molecular timing channels, the diffusion process and the channel are affected by the symbol duration. Moreover, the symbol duration is not fixed in such channels. So how can we denote channel capacity to be in bits per second, which is similar to the conventional radio-based communication where the symbol duration is fixed? So how do you relate fixed symbol durations to variable symbol durations?
Right. So this is a nice question, and it sort of goes to the fundamentals of how information is defined for random processes. So you might think that for each channel in discrete time, so in other words, if you have a fixed step associated with, or a fixed time step associated with your communication system, you might relate the information capacity to channel uses. So in other words, each channel use you can send a bit or something, and then you can change the units from channel uses to seconds because each channel use takes a certain number of seconds. You can change the units that way. When the symbol duration is not fixed, you can still talk about bits per second. What you now have is you have a random process. So you have a random process at the input with potentially, well, it's hopefully it's still stationary, but perhaps it could have some sort of Markov dependence on the input or something like that. Information is still defined in terms of time steps, if it's a discrete time or even in continuous time. The units might still be seconds in terms of, like, if you think about the time derivatives might be taken with respect to seconds if the random process is defined in terms of time derivatives, like Ornstein-Uhlenbeck process, for example. So we can still talk about the entropy rate by relating it to a unit such as seconds, and therefore we can also consider the information rate by subtracting or by calculating mutual information appropriately, again, with the units being in seconds. So there's not necessarily any requirement for symbol duration to be fixed in order to calculate information in terms of bits per second.
Next question is on lecture 16. Have there been any wet lab experiments to confirm the feasibility of molecular communication?
The answer is yes. So some people have gone to the wet lab and done experiments at the micro scale on actual organisms. I'm thinking mostly of groups in Japan that have done this. So if you're familiar with the work of Professor Tadashi Nakano, he's done a lot of nice work. I think he's actually done some lab work on this. There are other groups in Japan that have done actually implemented things like lab-on-chip systems that do molecular communication. Having said that, the wet lab experiments are very difficult and expensive, and the kinds of engineers who are interested in molecular communication often don't have a lot of experience in wet lab. So there tends to be a very steep learning curve to get up to wet lab experiments that can confirm the theories that we're talking about. I'm sure there are other groups who have done similar work, but my impression of the wet lab implementations of molecular communication so far is that they're quite rudimentary. So in other words, just very, very, very simple transmissions of single bits from point to point, maybe a total communication session amounting to just a few bits, and very, very slow data rates. I actually saw, I think, a paper about 10 years old, where I think they were trying to estimate the information capacity of a certain signal transduction system using wet lab experiments. These are biologists who are doing this, and I think they came up with something like 1 bit per minute. So these kinds of experiments tend to be very slow, and one major issue in molecular communication right now, and I think we'll talk about this when we talk a bit about open problems, is that there's a giant gap between the theories that we're developing mathematically and the actual practice of nanoscale communication, not macro scale, but nanoscale. There's a huge, huge gap between what we think we can do theoretically and what we are actually capable of implementing in the lab.
Thank you. The next question is sort of related. How close are we to the actual implementation of a micro nanoscale molecular communication system? Perhaps we could interpret it in terms of an application. How close are we to an application? Maybe
right. So yeah, this is a good point. If we interpret this question as asking how close are we to actually doing something like this in the laboratory, we've done this for the past several years. How close are we to a practical application of the ideas that we're talking about? I would say we're probably about a decade away. It's not terribly close right now, and I think that there's a lot of lab work that would need to be done. Moreover, if you think about, okay, so in the first lecture, what we talked about was nanomedicine. So we gave examples of nanomedicine. It is very, very, very difficult to perform any kind of experiment in the human body simply because of the safety protocols and the approvals that are needed, the expense of such an experiment, the controls that are involved in such an experiment. So even if we had a very nice application running in the lab today, it would still be years and years before that application could potentially become a real therapy that had passed clinical trials and that had gone through all the experimental steps. I think this is appropriate. I mean, no one wants to put people at risk, but I think we need to be realistic about the fact that development of medical therapies, if that's the goal, development of medical therapies is very, very slow. It's far slower than for example, the development of communication systems, conventional ones, or software, or things of that nature.
Thank you. Before we get to the next question, which seems like a planted question to get you to segue into new topics.
Yes.
Let's go to the Q&A. I think there are no questions on Q&A. Maybe I'll ask another question. So in the classical development of communication theory and information theory related to communication theory, there is the point-to-point communication, and then we think about multi-terminal information theory. You
Yes.
may have spoken about it in your talk, but can you highlight some interesting questions in that direction, where we are with respect to the MAC, the broadcast, and other such things? Are there some simplifications that molecular communications provides us, and therefore we might have some positive cases, or is it challenging as in the regular setting?
Yes, so there has been some work around layered design for molecular communication networks. There are important questions around things like routing and point-to-point transmission, ensuring that a particular message arrives at its destination. So there is actually one interesting technique that relies on things that we have talked about. And this was actually implemented. This is one of the things that I was thinking about as far as this question was concerned, as far as something that was actually implemented. So what they did was they had a molecular message, and what they did was they attached the message-bearing molecule to a so you know how DNA is two-stranded. There's two strands in the double helix, and they're complementary to each other. So what they did was they attached a single strand of DNA to the message, and that message was complementary to a strand at the destination, and basically this represented the address. So that if the message address matched the destination, it would actually bind, and the message would arrive at the destination. So there has been work in that direction. I think there is a lot of work to be done as far as the theory of for example, the network information theory of molecular communication. There's not been much done. That's very open. I also question whether our notion of layering that we have in conventional communication systems, if it is fully appropriate for molecular communications. I don't mean to say that layering itself is inappropriate, but I question whether a different kind of layering might be appropriate. So the challenges of molecular communication are very strong. We have ISI, we have very slow channels, we have very even poorly physically modeled channels. It seems like broadcast is a advantageous strategy if we look at natural communication systems. So I wonder if there's a different kind of layering that might be appropriate, and perhaps from a networking perspective, this might be an interesting direction of research.
Thank you. I don't see any other questions. In case you see some, perhaps you can take them.
Yeah, I don't see any on my board.
Yeah, if not, we can go to Adrish's question. What are the hot topics that can be explored in molecular communication field now?
This is a very appropriate question. I've actually prepared a few slides about this. Let me just get you over here. There we go. Right. So Adrish asked me to, I think he asked all the speakers to talk a little bit about open problems. And I think it should be clear from my lectures that there are a ton of open problems. This is a very active and growing field, and there's lots and lots to be done. So I broke my my open problems down into 3 areas, and these are by no means the only open problems. They are merely the ones that occurred to me first, probably because they're the ones that I personally find the most interesting. So, from an information theoretic perspective, there's actually not a lot known about molecular communication. And the headline question here is, what is the Shannon capacity of molecular communication? So if I ask you, what is the Shannon capacity of wireless communication? We have some fairly reasonable answers to that question. So we know that if I give you a bandwidth and a noise temperature, then you can calculate the Shannon capacity. It's bandwidth times log base 2, 1 plus SNR. There is no equivalent formula known for molecular communications. There's no even any reasonable method for developing such a formula in molecular communication. Moreover, if you think about how this question operates in wireless communications, it goes along with certain assumptions, such as bandwidth is limited, and there exists noise of a certain kind, and signal power is limited as well. In molecular communication, I think one major issue is that we don't even know what the right constraints are to apply to this problem. So in other words, things that we would normally assume are things that we might, initial assumptions that we might potentially make to apply to the Shannon capacity problem. They lead to either meaningless results or infinite capacities. So I think we touched on this a little bit in the lectures. I think we talked a little bit about what is the Shannon capacity per molecule, and it turned out to be infinite, or the Shannon capacity per second without constraints on molecules also turned out to be infinite. So this leads me to the question, how do we even approach the problem? What is a principled way in which to approach this problem? I think this is an extremely important problem because without knowing what the fundamental limits are, we don't know what the capabilities are. And my suspicion is that we are extremely far from the capabilities of molecular communication. So let me explain a little bit about why I think that. Currently, we have developed, as you've seen, experimental apparatus that transmit at a rate of, let's say, a bit per second. We've seen implementations in the lab, in wet labs, at microscale and nanoscale of a few, maybe at most a few bits per second. However, we have from my own results, we've shown that the information theoretic capacity of some kinds of signal transduction receptors can be on the order of tens of bits per second. So now we have an order of magnitude. On the other hand, we also have a result from my research that suggests that if you have, let's say, 100 signal transduction receptors on the surface of a cell, actually a more realistic number would be probably more like 1,000 signal transduction receptors on the surface of the cell, the information capacity goes up linearly, in other words, by a factor of 1,000. So now we've taken 10 bits per second times 1,000. This is now 10,000 bits per second. Now we're talking about a serious amount of information. This is like four orders of magnitude higher than anything we're capable of doing right now. And it can go up further from there. Like this could well be, if you think in terms of a systems biology sense, if you could exploit this in an engineered communication system, you could potentially get very high information rates. So it is natural to ask, what is the Shannon capacity of molecular communication? Is it small? Is it large? How do you even constrain it? What about even in specific circumstances? I mean, timing, we talked about this. This is a very hard problem to analyze, so maybe we just throw that out or put that aside for now. and we just ask what's the information theoretic capacity of concentration shift keying. As far as I know, that's unknown. I think that would be easier to solve, and that's something that would provide a very interesting result, I think. So the takeaway message from this slide is that we really don't know. We don't know what the Shannon capacity of molecular communication is. Even in nanoscale, fairly simple environments, it's reasonable to believe that it could be as high as megabits per second. And we're extremely far from achieving that. So there's huge, huge gains that are potentially realizable here. In terms of input, so there were questions about wet labs. I actually don't think the short-term low-hanging fruit, if you like, of molecular communication is at the micro and nanoscale. I actually think, as I mentioned in the last lecture there, I actually think it's in unconventional communication in what are called wave-denied environments. I think I provided a link to the paper. There is a very nice recent paper in, I think it's Communications Magazine, about wave-denied environments for 6G. So if we're truly serious about 6G being ubiquitous communication, then it has to provide a solution for places where waves, electromagnetic waves can't get, such as underwater, underground, or tunnels or metal structures. So we talked a little bit about those examples. There are commercially valuable and societally valuable applications where it would be really helpful if we could communicate with devices that were in those locations, underwater, underground, in applications such as infrastructure maintenance, search and rescue, things of that nature. Potentially, there could be commercial applications there that could potentially involve startups or more addressed markets that are valuable. So I think that is a nice short-term, very achievable goal. In terms of what you can do, and I'm addressing everyone now who's listening to this, especially students. What you may have noticed from my last lecture there in lecture 16, and I tried to make this point clear, is that I don't have a wet lab. I don't have any prospect of getting a wet lab because wet labs are expensive and I don't have the skills. I was never trained in wet labs. I mean, I could learn it, but it's difficult to gain those kinds of skills. So what we did was we started with just simple things that we could do by ourselves, and we were basically playing around with spare parts. It was very inexpensive. It's something that anyone can do. You can tinker with this in your house or wherever you are. It's not very expensive. You can get off-the-shelf components from whatever website you like. You can use processing components like Arduino. Very, very simple stuff, not very expensive, easy to do by yourself, and you can make a significant contribution to molecular communication research by designing and building these very simple apparatus that are not nanoscale at all, but again, might address some of these interesting wave-denied applications. One other thing that I like to think about is making use of the unique features of molecular communication. So what do I mean by that? One really neat thing about molecular communication is that it's persistent. So electromagnetic waves are not persistent. Once you emit them, they travel at the speed of light and they're gone. Molecular communication, what you could potentially do is you could leave a marker, and that marker will last quite a long time. And sometime later, another device could come by and read that marker. So you can imagine a fleet of robots leaving chemical markers for each other, so that they could leave each other persistent messages like tags or basically like graffiti, except in communication. So that's another idea for a potential implementation. Again, I don't think this would be terribly difficult to do for a clever student who is good with tinkering with small devices. I think you could easily produce something that might actually have an important application. Now, this is an area that I have started to get very interested in, which is molecular communication in biology. So as a sub-question of what we talked about in the first slide, we don't know what the molecular or what the information theory capacity of signal transduction is, or excuse me, of molecular communication is. We also don't know what the information theoretic capacity of signal transduction is, except in some specific examples. So we have actually solved some of this problem, but again, I suspect that the true value of the information theoretic capacity of signal transduction is fairly high. And it would be interesting to be able to exploit that and understand what the body is doing with all that information. As far as other applications of information theory and communication theory in biology, it has been noticed that the mutual information of molecular communication is related to fitness. So by fitness, you can mean many things, but for a bacteria, for example, the fitness is usually considered to be the growth rate. So a question is, can this be used to make useful predictions about biology? In other words, there seems to be some idea that if a sensing system, so let's say I'm sensing the environment, say I'm a bacteria, I'm sensing the environment, and the environment can either be good or bad. So if the environment is good, then I get out there and I divide as much as possible and I try to increase my growth rate. If the environment is bad, maybe I sort of shut all my systems down and I just go into survival mode and I try to survive the drought, or whatever you might call it. So there you can see that there's a sensing problem there. If I sense the environment and I think it's good, but it's actually bad, and I start to divide, then I'm almost certainly going to die. So I'm using all these resources incorrectly, and then I can't make them up by eating all the food that I thought would be there, then I'm almost certainly going to die. So you can see that there's an information component to fitness. And in fact, many biophysicists have noted that this does seem to be related to the mutual information. So a really neat question is, could this be used to make predictions about biology? If so, I think the implications would be profound. Then we would be able to say that Shannon's theorem is not only an engineering guideline, it is actually a law of the universe, and biological organisms have discovered it and are implementing it. Finally, can we apply these principles to analyze any biological system with a communications component? So that is related to this question over here. Many systems in our body have a sensing component that could potentially be interpreted using information theory. One example would be the immune system. So there is a process of discrimination by components of the immune system in which they have to tell the difference between a cell that is your own part of your own body, or a cell that is foreign that needs to be eliminated. So this is a process that happens all the time within your body. It's an incredibly good process because the immune system is immersed in objects that are of your own body. So I mean, your body is full of your own cells, and very, very rarely it comes across a foreign body, and it has to correctly distinguish those all the time. It can't. That process is not very tolerant of error at all. So a question would be, can we apply this kind of principle? Can we apply information theory to apply to analyze such systems? So those are basically the open problems that occurred to me that I could talk about in 10 minutes. I think there's lots more. I think there's tons and tons of stuff to do in molecular communication. And with that, I think I will conclude.
So let's all thank Andrew for his and also for taking the time to answer the questions that we have posed to him. Thank you very much, Andrew. And it was a pleasure to listen to your lectures as well as your responses to the questions. over to Adrish now.
Thank you. And
Yeah, Andrew, you were saying something.
I was just going to say, this has been a great pleasure to lecture to you. I'm extremely impressed with the quality of the questions and response. And I hope anyone who's interested will contact me and follow up with any questions by email. I'd be very happy to answer. So once again, this has been a huge pleasure. Thank you to the organizing committee and to you for organizing. Thank you for Josh for sharing today. This has been absolutely wonderful.
And thank you for accepting our invitation and giving wonderful lectures. It was really, I think, well received, and thank you so much. And hopefully we'll see you someday on our campus too.
Yes, I hope so too. I hope so too.
Okay.
Thank you.
Thank you.